Subject-specific vocabulary. Reaction rate. Reaction rate measures the speed at which a chemical reaction occurs and is defined as the change in concentration of reactants or products over a specific period of time. Reaction rate is expressed in molarity per second or moles per liter per second. What is an enzyme? An enzyme is a globular protein that acts as a biological catalyst, accelerating chemical reactions without being consumed in the process. All enzymes are proteins, but not all proteins are enzymes. As all proteins, enzymes have their own precise shape, their 3D structure, which is determined by their amino acid sequence. In other words, by their primary, secondary, tertiary, and/or quaternary structures. Destroying protein's native structure leads to destruction of its function. In other words, no structure, no function. No function, an enzyme is not working. It has undergone denaturation. Enzymes are biological catalysts. It means that the enzymes speed up metabolic reactions which are taking place in a living organism. Without enzymes for these reactions to form spontaneously products would take a lot of time. However, with enzymes, these reactions take place super fast. So enzymes increase rate of metabolic reactions by lowering a reaction's activation energy. So how an enzyme catalyzes the breakdown of a substrate molecule into two product molecules without being changed during the reaction? First, an enzyme has a cleft in its surface called the active site, where a substrate molecule with complementary shape can bind. Second, all molecules possess kinetic energy, which make them randomly vibrate or move around. This random movement of both the enzyme and the substrate bring them into collision, into a contact. If contact is tight enough, then an enzyme-substrate complex is temporarily formed. In this complex, R groups of amino acids in the active site interact with the substrate. So no tight contact, no enzyme-substrate complex. No enzyme-substrate complex, no product or no products. Third, the brief interaction of the substrate with the active site breaks the substrate apart, forming the product molecules. The products leave the active site, leaving the enzyme molecule unchanged and ready to bind with another substrate. So, are there factors that affect enzyme reaction? In other words, are there factors that affect reaction rate? Of course. First factor that affects enzyme action is temperature. At low temperatures, the reaction takes place very slowly, as decreasing temperature lowers molecular kinetic energy, making molecules move slowly. The result of it, collisions or contact between enzymes and substrates are also lowered. Less contacts, little products are formed. At high temperatures, the reaction takes place fast. Increasing temperature increases the kinetic energy of the molecules, so the enzymes and substrates move faster and collide with each other more often, forming more contacts. More contacts, more enzyme-substrate complexes. As a result of it, more products. However, above a certain temperature, the enzyme's kinetic energy increases so much that some of the bonds holding the enzyme molecule in its precise shape begin to break. Particularly, hydrogen bonds break first. The enzyme's active site begins to lose its shape, and therefore its activity. It is denatured. Second factor that affects enzyme action is pH. All enzymes have their own optimal pH conditions to work best. Some work fastest at low pH, most, however, at neutral pH. Nota bene, pH is a measure of the hydrogen ion concentration in a solution. So at low pH, the concentration of hydrogen ions are high. Positive charge on hydrogen ions interact with negatively charged R groups of an enzyme's amino acids, leading to its 3D shape change. At higher pH, the concentration of hydrogen ions are low, but concentration of hydroxide ions can be high. Negative charge on hydroxide ions interact with positively charged groups of an enzyme's amino acids, disrupting its structure. As a result of it, enzyme can make no contact with its substrate. No structure, no function, no product. So, beyond optimal pH, the enzyme activity first reduced, then reaction stops. Third factor that affects enzyme action is enzyme concentration. Increasing the amount of enzyme also increases the frequency with which the enzyme and substrate collide. As a result, enzyme-substrate complexes form more quickly, and the rate of reaction increases. However, there is a limit, as eventually there will be more enzyme molecules than substrate molecules. Some enzymes become excessive, as they do not have any substrate to bind. Any further increase in enzyme concentration has no further effect on the reaction rate. The fourth factor that affects enzyme action is substrate concentration. At fixed amount of enzymes, increasing substrate concentration also increases the initial rate of reaction. Because more substrates can make more collisions with enzymes' active sites. However, at one point, adding more substrate will no longer increase the reaction rate, because all enzyme molecules are already working at their maximum capacity. Enzyme active sites are full. Enzymes reach their Vmax. Vmax is the maximal reaction rate or velocity of an enzymatically catalyzed reaction when the enzyme is saturated with its substrate. Michaelis Menten kinetic is the model used to describe the enzyme kinetics in the enzyme catalyzed reactions involving the transformation of a substrate into a product. Michaelis Menten equation describes the velocity of enzymatic reactions by relating it to concentration of a substrate. Half Vmax is the substrate concentration at which half of the maximum velocity is reached. Km, or Michaelis-Menten constant, is the enzymatic kinetic constant that measures the affinity of enzyme towards the substrate. In biochemistry, affinity is a measure of the strength of the attractive force between two molecules, such as receptor and its ligand, or in our case, an enzyme and its substrate. Why do we compare enzyme affinities to its substrate? The maximum rate of reaction Vmax is used to derive the Michaelis-Menten constant. This is then used to compare the affinity of different enzymes of their substrates. A high affinity, that is a low Km value, means that an enzyme makes contact to its substrate strongly, thus increasing product formation. The higher the affinity of the enzyme for the substrate, the lower the substrate concentration needed to reach half Vmax. The lower the value of Km, the more efficient the enzyme. If the affinity is low, the substrate may leave the active site before a reaction takes place. The higher the value of Km, the less efficient the enzyme.